Now we're looking at the reciprocal control of glycolysis and gluconeogenesis. Very quickly, glycolysis is a catabolic process. Gluconeogenesis is an anabolic process. And so, in order to have efficient running of the cell, we have to perform reciprocal regulation. And this reciprocal regulation ensures only one pathway is running at any particular time to avoid a futile cycle. Futile cycles happen when complementary or reciprocal pathways run together in the same location at the same time. We achieve nothing, no net change, and all we do is waste energy. Here are the learning objectives, which you should read through on your own. We are going to focus on the 3 key points for regulation or reciprocal regulation of glycolysis and gluconeogenesis. So these 3 steps are the bypass steps. And they are step 1 of glycolysis, catalyzed by hexokinase, step 3 of glycolysis, catalyzed by phosphofructokinase 1, and you always got to say 1 because there is a phosphofructokinase 2 that you're going to see in a couple minutes. And then step 10 of glycolysis, which is catalyzed by pyruvate kinase. More learning objectives, and these are specific to each enzyme. So I want to draw in PFK1 here. Take a moment to read through these. Beginning now with hexokinase. Hexokinase is found in all of the tissues of the body. Glycolysis is very highly conserved evolutionarily, and for a cell to be viable, meaning living, it needs to have some type of glycolysis occurring. Hexokinase is found in all tissues, and there are multiple isoforms of hexokinase. The liver has hexokinase isoform 4. So the liver has hexokinase 4, and this is also known as glucokinase. How are hexokinase in all of the other body tissues and glucokinase, aka hexokinase 4 in liver, different? Well, hexokinase is inhibited by glucose 6-phosphate. Glucose 6-phosphate is a product of this reaction. And so most of the hexokinase enzymes in the body are inhibited by glucose 6-phosphate. So they're sensitive to product inhibition. Glucokinase in liver is not inhibited by glucose 6-phosphate. So we can avoid product inhibition in liver. This means that hexokinase 4 in liver will continue to form glucose 6-phosphate even when there is a high level of glucose 6-phosphate present in liver cells. Hexokinase in most of all of the other body tissues is a constitutive enzyme. Constitutive enzymes are always expressed. So the gene is always on, and we're always going to be forming enzyme at the ribosome, and hexokinase enzyme will always be active in all of the body tissues. Glucokinase in liver is induced by insulin. An inducible gene experiences increased transcription when the activating compound is present. So insulin increases transcription of the hexokinase gene in liver, which is hexokinase 4 or glucokinase. And we're going to have also increased translation of the mRNA to protein at the ribosome, And so we will have increased hexokinase 4 activity in liver. So insulin induces or increases the activity of hexokinase 4 in liver. Hexokinase in all of the other body tissues has a very low Km for glucose. Now, I'm simplifying this for you because you didn't have Biochem 1, which is where we cover what exactly is a Km. You can, although it's not perfectly true, you can think of a Km value as representing the affinity of an enzyme for a substrate. And so if we have a low Km, we have a high affinity. And that means that the enzyme will use the substrate when it's present in very low concentrations in the cell. So there will be a high activity of the enzyme even when there's a very low amount of the substrate. Let me sum this up. All of the other body tissues have first, let me erase some of this. All of the other body tissues, not liver, has first dibs on glucose. So everybody else gets to use glucose first. Liver has last place, right? So everybody eats first and the liver eats whatever's left. So glucokinase in liver has a high Km for glucose. This means, right, a high Km in general, and again, it's not perfectly correct, and I cringe to even write it, but a high Km means that we have a low affinity of the enzyme for glucose, and so we're not going to have activity of the enzyme until the substrate is at very high concentration. So enzyme activity, or enzyme is active only at high glucose concentration. This means that the liver has last rights, right, at glucose, or last chance at glucose. So what does this mean? The liver is very altruistic. The liver permits muscle and brain to have glucose when blood glucose levels are low, and the liver will refuse to use or trap glucose under those conditions so that muscle and brain can eat first. Here is a graphic representation of this property. So on the y-axis, we have relative enzyme activity, and you could think about this here as 100% activity. And down here is, right, of course, 0% activity. Here you can see glucose concentration in millimolar. Think about this as glucose concentration in blood. Hexokinase 4 in liver has a very high Km for glucose, and so it only is active at high glucose concentrations. So if we look here at 5 millimolar glucose, which is about where blood glucose normally hovers, there's not a lot of activity of the hexokinase 4 in liver, but you can see that hexokinase 1 in non-hepatic tissue is very active. This enzyme has a low Km and high affinity for glucose. Okay, this pertains to hexokinase 1. Hexokinase 4 has a high Km and low affinity for glucose. Regulation now of phosphofructokinase 1, PFK1. Well, we have a PFK2, so you always got to say PFK1. So shame on me for not having it there. PFK1 catalyzes the committed step of glycolysis, and commits the carbons of glucose to glycolysis. And that's because fructose 1,6-bisphosphate really has no other metabolic pathways to flow into in the cell. So we commit those carbons then to glycolysis. PFK1 is allosterically inhibited by ATP. So think that glycolysis is shut down in high energy conditions within the cell. PFK1 is also allosterically inhibited by citrates. So again, glycolysis is inhibited in high energy situations in the cell. So when we have enough ATP, when there's high levels of citrate because the TCA cycle can't run fast enough, we then have enough ATP and we don't need to any longer run glycolysis. So we'll shut down PFK1. PFK1 is allosterically activated by fructose 2,6-bisphosphate. This is an indicator of low energy within the cell. And we will then activate PFK1 to activate glycolysis to make more ATP because the cell needs more ATP. Allosteric regulation, right? So just allosteric regulation involves the non-covalent reversible modification of an enzyme by a small molecule. And this causes a shape change in the enzyme to increase the affinity of the enzyme for binding substrate. We're going to look at it. Here you see a graph of PFK1 activity under conditions of high ATP and low ATP. This is the amount of substrate for the enzyme. So when there is not much ATP present, we have high activity of PFK1 when there's not much substrate present. So the PFK1 is very active in low ATP or low energy conditions. When there's a lot of ATP present, you can see that even at higher substrate concentrations, at high fructose 6-phosphate concentrations, there's not much activity of our PFK1. And so when there's high ATP, PFK1 is not very active. And so the flow through glycolysis is responsive to the energy needs of the cell. Here you can see something that was looked at in your homework, and we're looking at a glucose analog, a molecule that mimics the structure of glucose, except instead of an OH, we have a fluorine. So this was OH in glucose. Now, this molecule is used as a radio tracer in positron emission tomography studies to look for locations where there are very active cells in the body, meaning cells that are using a lot of glucose in the body. And if we couple a CT image with a PET image, right? We have here, this is PET CT. When we overlay those images, we can very clearly see locations of metastases in the body. Now, here we've formed 6-phospho-2-fluoro-2-deoxyglucose, and what happens to this molecule next? Does anything happen to this molecule next? You have to look at the mechanism of the phosphohexose isomerase enzyme to see what may or may not happen to this molecule next and why. Phosphofructokinase 1 is like hemoglobin in that it has 4 subunits. It's sensitive to allosteric effectors like ATP, AMP, citrate, etc., and intersubunit communication. So basically, we have 4 subunits here. Each subunit is shown to you in a different color. If we have binding of an allosteric effector, so binding of a small molecule like ATP to one of these subunits, it's going to cause a shape change in that subunit where it binds, and then that shape change is going to be communicated to each of the other remaining subunits. So it's a very sensitive enzyme to allosteric regulation. This PFK1 enzyme, which we do cover in Biochem 1, is like a big thick donut. And the allosteric effectors like ATP, citrate, AMP, and this fructose 2,6-bisphosphate bind within this central hole in the enzyme. And then we have the catalytic sites, which are converting fructose 6-phosphate to fructose 1,6-bisphosphate, are located more towards the outside of this enzyme complex. Here you can see a schematic representation of the inactive form of the enzyme, also known as the T state, and the active form of the enzyme, also known as the R state. We refer also to T and R states in hemoglobin, where R states in hemoglobin have high oxygen binding affinity and T states of hemoglobin have low oxygen binding affinity. PFK1 is an important integration point for metabolic signals. And so PFK1 is also sensitive to high citrate. And citrate is the first intermediate of the TCA cycle. And so when TCA cycle is going slow, citrate levels increase. And those high citrate levels can feed back to shut down the running of glycolysis, which makes pyruvate, which is going to flow into TCA cycle as acetyl-CoA. And we will come back to that when we look at TCA cycle. For right now, take a quick look at this graphic. We have PFK1 activity on the y-axis, and we have fructose 6-phosphate substrate concentration on the x-axis. Consider just this amount of fructose 6-phosphate. So we're only going to consider along this line. When we have no inhibitors present, meaning low ATP, or again, low energy state within the cell, we have the highest level of PFK1 activity. And this is because we have low ATP. We need to make more ATP, so we got to run glycolysis. And so we're going to be replenishing ATP in the cell. We have very high PFK1 activity. PFK1 runs the committed step for glycolysis. If we have high energy within the cell, we have low PFK1 activity. We got a lot of ATP. We don't need more ATP, so why don't we shut down glycolysis? Look what happens on this red line in the middle here. We have the same amount of ATP as we did in the high energy state, but now we have just 1/10 the amount of ATP present as AMP. We have 0.1 millimolar AMP, and AMP is a low energy signal. And AMP is a very strong activator of PFK1. So when we start to have even a small hint that the cell is entering into a low energy condition, we will greatly upregulate the activity of PFK1 and start to run glycolysis at a high level. So the cell anticipates this coming low energy state and will start to run glycolysis in order to maintain the proper needed amount of ATP within the cell. On a molecular level, and we're not going to cover this because this is Biochem 1, we have this T to R structural transition, right? So we're going from inactive to active, and there is actually, honest to goodness, like this little switch within the enzyme. So we're going to flip the positions of two amino acids. One is positively charged and one is negatively charged. And what essentially we're doing is increasing or decreasing the affinity of PFK1 for fructose 6 phosphate, which is shown here. And that is the substrate for PFK1. So you can see here in the inactive state, we have glutamate 161 present. Glutamate 161 present in close proximity to the active site is, right, this is negatively charged. The substrate's negatively charged. Like charges repel. We're going to decrease binding of substrate. When we have the R state, we have arginine 162 present in close proximity to the phosphate group on fructose 6 phosphate at the active site. And so opposites attract. We increase the binding of our substrate and we increase the activity of the enzyme. Fructose 2,6 bisphosphate is an essential regulator of glycolysis because it is an allosteric activator of PFK1. And where do we get this fructose 2,6 bisphosphate from? We get it from the action of PFK2, right? So phosphofructokinase 2. And phosphofructokinase 2 enzyme is going to add a phosphate to carbon 2 of fructose 6 phosphate, right? PFK1 adds a carbon. PFK1 adds a phosphate to carbon 1 of fructose 6 phosphate. So right now we're focused on PFK2, and we can make fructose 2,6 bisphosphate. Now, to get rid of fructose 2,6 bisphosphate, there's an enzyme called fructose 2,6 bisphosphatase, which removes a phosphate and will deplete the level of fructose 2,6 bisphosphate. Phosphofructokinase 2 is active when we need to run glycolysis. Fructose 2,6 bisphosphatase is active when we need to run gluconeogenesis and, right, does this by depleting the level of fructose 2,6 bisphosphate. So we're going to reduce the running of glycolysis through, right, decreasing the level of fructose 2,6 bisphosphate, which decreases the activation of phosphofructokinase 1. Now, back to some graphs of activity of phosphofructokinase 1. So here we're looking at activity of phosphofructokinase 1, right? Both graphs. And here we're considering the amount of fructose 6 phosphate. If you look at the red line here, the red line is saying that there is no fructose 2,6 bisphosphate present. And so we have very, very low activity, right? So here's the activity, very, very low activity of our phosphofructokinase 1. If we add just a very small amount of fructose 2,6 bisphosphate, we get a huge increase in the activity of our PFK1. So PFK1 is strongly activated by fructose 2,6-bisphosphate. In fact, we need a tiny bit, like 0.1 micromolar. That's a crazy small amount. We need a little tiny bit, right? So this is 0.1 times 10 to the minus 6th molar, which is 1 times 10 to the minus 7th molar. That's a very small amount of fructose 2,6-bisphosphate. We need a little bit of fructose 2,6-bisphosphate in order to have any appreciable activity of phosphofructokinase 1 within the cell. So if we deplete the level of fructose 2,6-bisphosphate through the running of fructose 2,6-bisphosphatase, we are going to really shut down the activity of phosphofructokinase 1 and therefore shut down the activity of glycolysis. Fructose 2,6-bisphosphate is a very potent activator of phosphofructokinase 1, and fructose 2,6-bisphosphate greatly overpowers the inhibition of PFK1 by ATP, and that's what you're seeing here. So if there is no fructose 2,6-bisphosphate present, even a small amount of ATP is able to shut down the activity of our PFK1. So note that this is in micromolar, so 10 to the minus 6th micro. If we add a tiny amount of fructose 2,6-bisphosphate, so 0.1 micromolar, we're able to relieve some of that inhibition of PFK1 activity by that same amount, right, present of ATP. So we're considering what's happening here at 2 micromolar ATP. If we add a bit more fructose 2,6-bisphosphate, we greatly increase the activity of PFK1. So fructose 2,6-bisphosphate is able to strongly activate PFK1 even in the presence of ATP.